Hello, my name is Rebecca Pantoja, and today I'm going to talk about G protein coupled receptors. First, let's get into what makes up a G protein receptor. It is made of three monomers such as alpha, beta, and gamma, and it is found in only eukaryotes such as humans, plants, animals, and even yeast. They are considered 7 transmembrane receptors because they are capable of passing through the cell membrane 7 times. So let's get into their specific function or role in the cell. They become essential when certain signals need to pass through the membrane, which allows for a conformational change within the G protein, and this will allow activation of the second messenger in the cell to activate other proteins via phosphorylation. Certain molecules may be too large or their chemical structure may not allow them to pass through. For instance, if they are polar, such as charged molecules, they will not be able to diffuse through the membrane, and even hydrogen ions are not able to freely diffuse. Some examples include hormones, pheromones, and neurotransmitters. And so, as I mentioned earlier, the G protein receptor can involve different subunits, alpha Q subunit or alpha S unit. Depending on which subunit is attached to the protein, it will determine the activation of a specific second messenger. If alpha S subunit is attached to the protein, this will allow adenylate cyclase to produce cyclin AMP and allow the second messenger to activate protein kinases in the cell. This will trigger a response to stimulate cellular secretion or open ion channels to allow sodium or other charged ions to enter in a high concentration, allowing a reversal membrane potential within the cell. This is just an example. There are a lot more, such as if the subunit is alpha Q, membrane bound PLC being the effector, it will release IP3 and DAG, also known as diacylglycerol. Excuse me, I can't pronounce that. These are the second messengers. Furthermore, DAG will activate PKC and cause phosphorylation of other proteins in the cytosol. Basically, phosphorylation is just adding a phosphate group to a molecule. This will then lead to a cellular response, such as cell contraction or secretion. And again, these are just few examples about how subunits can affect which second messenger is activated. But there are a lot more, such as alpha 12 and alpha I. So finally, these diagrams basically demonstrate the mechanisms in further detail of both alpha S and alpha Q and how they both work in the cell. The diagram on the left exemplifies a hormone coming in contact with the cell membrane and binds to the G protein. You then can see how there is a change from GTP to GDP. This mechanism does require ATP in order for the adenylate cyclase to activate inactive kinases and allow for the response to be triggered. The diagram on the right shows the messenger coming in contact with the receptor at the membrane, same as the other one. This then allows the subunits to dissociate, and IP3 and DAG act as the second messengers, and then they can allow for PKC to phosphorylate other proteins until it reaches the cellular response, and then cause a cell contraction or secretion. And again, if a GPCR alpha subunit is an alpha S, you can see that cyclin AMP will be produced. If the subunit is an alpha Q, phospholipase C, also known as PLC, will then be activated in the cell. So in conclusion, when a ligand binds to the GPCR, such as a hormone or neurotransmitter, it causes a conformational change in the GPCR. This will allow it to act as guanine nucleotide exchange factor, also known as GEF. The GPCR can then activate an associated G protein by exchanging the GDP bound to the G protein for a GTP. The G protein's alpha subunit, together with the bound GTP, can then dissociate from the beta and gamma subunits to further affect intracellular signaling proteins, or target functional proteins directly depending on the alpha subunit type, whether it is alpha Q or alpha S, alpha 12 or alpha I. And that concludes my my slideshow on G protein coupled receptors. So thanks for watching.